Hello, this is the third talk within the epigenetics session. In this talk, I will talk about microRNAs in skin homeostasis and development. In the first two parts in this session, I have introduced you to the expanding RNA word, and also in particular to the word of microRNAs. Why do we have them? How do they work? And why do we have so many? In this part, we will zoom in on to the skin, which is the organ of interest for all of us, and see what microRNAs are doing in the healthy skin. In this talk, I will address the following questions. What is the role of microRNAs in the skin? What happens without them in the skin? And how do microRNAs regulate epidermal differentiation in health and disease? So you may remember that in the previous talks, I talked about the importance of microRNAs for complex organisms like humans, like us. And also, I showed you some of the mouse experiments which have been done, in which enzymes or proteins necessary for microRNA processing were deleted. So, and in this case, these mice did not have any microRNAs, or almost all microRNAs need these enzymes for their processing. And what happened? These mice did not make it very far. So they were not viable. They did not make it further than embryonic day 6.5 to 9.5, when Dicer, Drosha, or EGO2 was deleted. So this is what happens if we remove the microRNAs in the whole body. But what happens if we remove them specifically in the skin? Can skin development go on normally without microRNAs? To address this question, a very similar approach was used in the year 2006, first time. So Dicer, the enzyme, one of the enzymes which is needed for the processing of almost all microRNAs, was deleted specifically in embryonic skin progenitors. And this study was published 2006 and was performed by the Fuchs group. And what happened to these mice? Well, they also did not make it very far. So they died a few days after birth, 4 to 7 days within the first week of life. And that was due to severe dehydration. And when their skin was examined more closer, there were several histological abnormalities observed. What was mostly apparent that their hair growth was deficient, that the hair follicles were upside down, basically. Instead of invaginating, they were evaginating upwards. And what could also be observed that also the interfollicular epidermis showed some abnormalities. There was an increased epidermal proliferation, and there was also a decreased number of granules in the granular layer. So that was what happened. And also when DCGR8, another gene which is coding for an enzyme needed for microRNA processing, was deleted, it had very, very similar effects. So what is the conclusion? Mice are not viable without an intact microRNA machinery in the skin. So at this time, these experiments were really important to show the role of microRNAs in skin development, but they did not give any information about the role of specific microRNAs in the skin. And it is also possible that some effects were not detected, because if you think about some microRNAs can increase proliferation and some can inhibit it. If you delete both of them, maybe you don't even see any difference. So the next step was to find out what happens if individual microRNAs are deleted. And there have been, these are just few examples for some of the microRNAs which are expressed abundantly in the human skin, and in particular in the epidermis, and which have been modulated either by knockout or by generation of transgenic animals overexpressing these microRNAs specifically in the epidermis, or by inhibition of these microRNAs by injection of antagomeres. And you can see that all of these showed a phenotype and the epidermal development or the hair follicle development was disturbed. This is a figure showing some of these microRNAs which are important in the development of the skin, in particular miR203 and miR24 in the epidermis, regulating proliferation and cytoskeletal rearrangement, respectively. miR205 is not mentioned here, but it was also important in the epidermis. But this microRNA is also important in hair follicles, as well as miR125B is also needed for proper development of hair follicles. And the miR125B was also regulating the proliferation of sebaceous glands. So these are some examples, and there are definitely more microRNAs which are important for the proper development of the epidermis and the hair follicles. So I would like to talk a little bit more detail about one microRNA, which is important in skin and epidermal development. And this is, of course, not the only one, far from that, but this is a very nice example for a microRNA which is important in healthy skin. And as you will see, it has also relevance for some skin diseases. So miR203 is a microRNA which is enriched in the skin. And we bumped into this microRNA accidentally when we were investigating the microRNA expression profile of the inflammatory skin disease psoriasis and atopic dermatitis, and compare them to each other as well as to healthy skin. And we identified miR203 as a microRNA which is upregulated in psoriasis skin. And then we investigated it further together with some other microRNAs. And as you see, there is a difference between the expression pattern of these microRNAs. We investigated their expression pattern in different organs and tissues to see where they are expressed. As I mentioned before in the previous talks, some of the microRNAs are organ, tissue type, or developmental stage specific. And what we saw that most of these microRNAs that we identified were expressed in several organs, but miR203 showed a very specific expression pattern. It was expressed in the skin and in the esophagus, which also contains squamous epithelia. So we hypothesized that this microRNA may have an important role, specific role in epithelium. But the skin, of course, is a complex organ with different cell types, a complex tissue. And therefore, as a next step, we investigated whether miR203 is expressed in only one or several cell types within the skin. And you can see this was the result of this experiment, which showed that miR203 is expressed specifically in the keratinocytes, a little bit of expression in some melanocytes, but otherwise not in fibroblasts and not in any immune cell types that we investigated, not in mast cells. So it seemed to be that this is a keratinocyte-enriched microRNA. And the next step was to look at its expression in vivo. So these were like cultured keratinocytes, but in vivo, the keratinocytes form this complex three-dimensional structure, the multi-layered epidermis, in which they differentiate and continuously move upwards. And this process is very tightly regulated. So we investigated the expression pattern, the spatial expression pattern of miR203 in healthy skin using in situ hybridization technique. And what we saw is, if you look at the purple color, which indicates miR203 expression, that it was present mainly in the suprabasal layers and absent or almost absent from the basal layer of the epidermis. And we thought that this expression pattern was very interesting, because it led us to the hypothesis that is it possible that miR203 could have a role in the transition from the basal layer to the suprabasal layer in the process of differentiation. And indeed, another group, Elaine Fuchs' group, they showed a very similar expression pattern also in the mouse skin. As you see, it was miR203 was also absent or almost absent in the basal layer, with a high expression in suprabasal layers, also in the murine skin. Also they could show a very specific expression in the epidermis. And interestingly, this microRNA is only present in vertebrates and only in organisms which have a stratified epithelia. So in this Nature paper from 2008, they also showed that forced expression of miR203 in the epidermal progenitors suppressed the proliferation and induced differentiation. So we did this microRNA regulated the differentiation of the keratinocytes. So how they did that is that they expressed miR203 using the keratin 14 promoter, specifically in the epidermis, and then they express it in the basal layer where this microRNA normally would not be expressed. And the result was a thinner epidermis, and also the expression of the early expression of differentiation markers such as K10 already in the basal layer where it shouldn't be. So the cells started to differentiate earlier than they should. And at the same time, the expression of stem cell markers such as P63 was decreased. So altogether, these results show that miR203 is a microRNA which is expressed specifically in the skin, specifically in the keratinocytes, and suppresses epidermal proliferation and promotes the differentiation of the keratinocytes. And it is very important for skin development and for the normal homeostasis of the epidermis. And why is this interesting? Partially, of course, for skin biology, it is very important. But this balance between stemness, proliferation, and differentiation is important not only for healthy skin, but also when it gets disturbed, it can lead to different diseases. And in many skin diseases where the epithelium is affected, both inflammatory skin diseases and malignant skin diseases, this process of epidermal differentiation is severely disturbed. It also means that regulators of this process may be involved in the pathogenesis of these diseases. Aging, which is in itself not a disease, but it is also associated with disturbances in the epidermal homeostasis, is also a process where one can hypothesize that regulators of epidermal differentiation may be changed and they may have a role in skin aging. So back to miR203 again, because this is an interesting microRNA, and we have shown here so far that it is an important microRNA for regulating the balance between stemness and differentiation. And it is a very strong promoter of epithelial differentiation. So what happens then when this epidermal differentiation is disturbed, for example, in malignant skin diseases? These data I show here are from the group of Andor Pivarci, and I have been involved in those myself as well. So in basal cell carcinoma, which is a keratinocyte-derived skin cancer, we investigated the expression of this microRNA, and we found that its expression is suppressed or almost absent in these tumor cells as compared to the normal skin or the normal epidermis. And this is actually maybe what you would expect, because these cells do not differentiate normally. They are kept in an undifferentiated state. In squamous cell carcinoma, the situation is a little bit more complex. It is also a keratinocyte-derived cancer, but it can have different differentiation grades. And what we saw here is that in well-differentiated cancers, the levels of miR203 were not changed significantly. However, it showed a decreasing level of expression with lower differentiation grades. So cancers with a poorly differentiated grade, they had lower levels of miR203. So what would happen if we would substitute this missing miR203 from the tumors? Would that have any effect on the tumor growth? And this is the question that we asked. We performed animal experiments to answer that with tumor cells, squamous cell carcinoma cells overexpressing miR-203. So these cells, as well as control cells, were injected into immunodeficient mice in a xenograft model. And what we saw was that forced overexpression of miR-203 in the squamous cell carcinoma cells led to smaller tumors. They had both lower weight and lower tumor volume. So this led us to this model that low miR-203 levels in the basal layer of the epidermis, as well as in cancers, they can contribute to the undifferentiated phenotype of transformed cells. And also in the tumors with already established keratinocyte-derived tumors, low miR-203 levels can contribute to the growth of transformed cells. And if we overexpress miR-203, it could suppress the tumor growth. So altogether, these and other results have led to the following model. This beautiful picture is from Andor Pivarci. In this model, this is the model to describe the role of miR-203 in skin homeostasis and cancer. And maybe you remember that I have showed you the bistable switch model, in which a microRNA regulates its targets and the target regulates the microRNA. And this is a beautiful example of such a bistable switch. So in normal conditions, calcium, which forms a gradient in the epidermis and induces differentiation. Calcium induces also miR-203, and miR-203 in turn suppresses the expression of, for example, c-Jun, but also of other known miR-203 targets, such as p63, MYC, and others. But with the c-Jun, it forms a bistable switch, because c-Jun also suppresses the expression of miR-203. So if calcium induces miR-203, that leads to low c-Jun levels, which in turn allow high levels of miR-203, which in turn further suppress c-Jun, and this leads to a one-way road of differentiation. So these cells do not turn back. This is a program that they follow. On the other hand, in malignant cells, in BCCs or SCCs, what can happen is the mutations in the hedgehog pathway in BCCs or in the EGFR pathways, typically in SCCs, induce c-Jun or other regulators of miR-203. If c-Jun is high, it suppresses miR-203, which allows high levels of c-Jun, which further suppresses miR-203. So the cells will be in the other state of the bistable switch with high c-Jun levels and low miR-203 levels. And the constantly low miR-203 levels and high c-Jun levels do not allow these cells to differentiate. And these cells are the cancer cells, BCC or SCC cells. So in this sense, miR-203 acts as a tumor suppressor microRNA in the skin. So in summary, microRNAs are involved in skin development. There have been many studies now with transgenic and knockout animals showing that microRNAs regulate epidermal differentiation, they regulate skin development, and also the development of hair follicles, and in some cases also the sebaceous glands. So microRNAs, some of them can also regulate the balance between stemness and differentiation in the epidermis. And these processes can be disturbed in different diseases. And accordingly, microRNAs have been implicated in several diseases of the skin. I have showed you one example here, miR-203, which is suppressed in both basal cell carcinoma and low and poorly differentiated squamous cell carcinomas. But there are many other microRNAs which have been shown to play a role in these keratinocyte-derived cancers. I will talk more about chronic skin inflammation in the next talk, but several microRNAs have also been shown to be differentially expressed in several chronic inflammatory skin diseases and contributes to the pathogenesis. And aging is also something that where several microRNAs have been implicated. Differential levels of microRNAs have been shown in the aging skin. Now I have focused a lot on the epidermis, but also, for example, in fibroblast senescence, several microRNAs are changed in their expression, and it has also been shown that in the aging human skin, microRNA expression is changed. So I think that there is still a lot to discover, but one take-home message is that microRNAs are involved in skin development, epidermal differentiation, carcinogenesis, and aging in the human skin. Thank you very much for your attention.